so let's get started. Oh, but just one. Okay. So last time, in the last class, we discussed localized versus distributed brain function, and most of the neuroscience research is pointing towards which one? Localized, right? So very specific functions of very specific areas. Right? Then we looked at MIT Neuroanatomy series and Secret of the Brain. Right? So today we are going to finish the Secret of the Brain and Future Devices and Homework 7, which is a map of the brain, and Introduction to fMRI data analysis. We'll start with fMRI. Anyone, any questions on the assignment 1? Did the tutorial happen? What is the deadline? Third, okay. So you are a little late. So you would have figured out anyway yourself. Okay. So you are comfortable with assignment 1? All set? What are you using, SPM or FSL? Huh? You are using SPM. How about you guys? Not yet started, huh? FSL? FSL? Okay. Right. Okay. So note down your homework 8. TED Talk by Nancy Kenweiser, A Neural Portrait of the Human Mind. So one of the course student, one of the alumni of this course, I think she was from cognitive science, did this same course, and then she went to the US or not somewhere, I guess US or somewhere in the Europe, and there she met Nancy Kenweiser. And he goes, sir, she sent me an email. Sir, I met Nancy Kenweiser the other day. And what do you talk to her about? We just talk general things. But yeah. So she is in Boston area. I think MIT. No? Is not yet on Gradescope. That's fine. We'll put it on Gradescope. We'll get time. We'll do it before next class. But you're supposed to read it by now. You're supposed to do it by now. It's a TED Talk by, I don't have the full citation here. It's called A Map of the Brain by, I don't remember. Can somebody remember who's giving this talk? Nobody has done it, huh? You didn't do it? No? Okay. I need to be more regular, that means, I have to post it, otherwise you guys won't do it. So then we will not discuss it today. If you have not done it, then how can we discuss it? We will discuss something else. Alan Jones, yes, you're right. Okay. So we looked at Capgras delusion, phantom limbs, synesthesia, blind sight, hemispatial neglect, right? We kind of covered all of these. Temporal lobe epilepsy is something that we did not talk about here. So we talked about phantom limb, we talked about different pathways, how pathway and what pathways, so you kind of are comfortable with that, right? I'll post a reference. No? Okay. I'll post another homework. It's a nice primer, Introduction to Neuroscience, and then I'll just post it. You can read it on your own time. But that could be a part of the course, okay? So, yes, temporal lobe epilepsy. Anybody has any comments on temporal lobe epilepsy?
Finding profound meaning in everyday things.
So, so what is the phenomenon?
Experiencing God. Experience of spiritual and God.
Yeah, but even before that, why did he go to Ramachandran? John. What was his name? John, right? So why did John go to Ramachandran in the first place? Yeah. So he was having epileptic seizures. Temporal lobe epilepsy. Well, he was having some seizures. And what does seizures do? So just walking, doing the normal thing, all of a sudden, your body will start jerking, different parts will start moving uncontrollably. That's a seizure, right? It's because of epilepsy. And then when he went to Ramachandran, he figured out that he has a temporal lobe epilepsy. Right? So seizures. And what do we mean by temporal lobe epilepsy?
Random firing of neurons in the temporal lobe.
But if you are having seizures, your hand is moving uncontrollably, your body is moving uncontrollably. What part of the brain is actually controlling that? Motor cortex, which is in the frontal lobe. Then why is it called temporal lobe epilepsy?
Because it is happening in the temporal lobe.
Temporal lobe is primarily involved, and from there it is actually the origin of the seizures, origin of the neural activity. So when you look at this, there are different types of epilepsies: focal, multifocal, and generalized. This. But anyway, so in one specific case where you can localize, you can say that the seizures are what are seizures? Basically, neurons firing uncontrollably in a different manner, in a different pattern than usual, and what happens is some areas, maybe for the focus epilepsy, some areas will start firing, and then those action potentials are going to spread. They spread throughout the brain, and that leads to these seizures. Right? That leads to these epileptic fits or seizures. And it is temporal lobe because they look at it. Maybe they put a cap, do the EEG analysis, and based on that they conclude that the origin of seizures is in temporal lobe. So temporal lobe would have the maximum neural thing. As you go away from the center, the epicenter of the seizure, there'll still be, the activity will still flow into those areas, but it will not be primary. It will not be as involved. Okay. So that is one part of the phenomena, and the diagnosis was that he has temporal lobe epilepsy, right? But interestingly, in addition to seizures, what are the other things that were observed for this patient, John? What did he experience?
He started believing he was God.
He started believing that he was God. Okay. And there is a very interesting dialogue, right? He says something, he parked the car in the wrong place or something, and then the neighbor came in and said that I'll call police on you. Right? And then he said that, you can't call the police on the God, right? So that's what the strong conviction was, right? He was God. What else?
It also mentions that they feel deep emotional connection.
Yeah, deep.
Atrocities happening all around the world and stuff.
Emotional connect and concern about atrocities, right? Or rather, I should say injustice in the world.
So he was mentioning that somebody is walking on a painting, reference to a painting he was doing. That one side it is happening joy, and on the other side of is facing the pain, excruciating pain and transcendent joy.
So joy and pain at the same time, together. Rapid mood swings, right? Mood swings were there, right? Sometimes he would either at the same time or in quick succession. Okay?
In between two episodes of seizures continuously, he was getting some thoughts, and he was correlating the thoughts with some spiritual activities.
Yeah. Some spiritual experiences, right? Like what?
Like prophetic insight,
Hmm?
insights he was mentioning.
Insights. What kind of insights? You remember? It's been a while I watched that.
He was omnipotent.
Huh?
He believed he was omnipotent.
He believed he was omnipotent. It's a different thing. Whether he believed it or he was actually omnipotent. Right? There are two different things.
He believed.
Believed. But was he? Okay. Hmm.
So he was not religious before.
Not religious before. Hmm. Okay.
And that one more thing was explained by Ramachandran that this is happening maybe because of pathway which is correlating sensory areas in temporal lobe and amygdala, due to which he is experiencing such kind of.
Okay, so what's the explanation of this phenomena, right? One possible explanation could be, let me say possible explanations, right? Pathway between
Sensory area and temporal
sensory and temporal lobe. Amygdala and temporal, right?
So he says that this connection gets strengthened, which causes increase in emotional saliency.
Amygdala and temporal lobe. So basically, your temporal lobe is firing, right? The neurons that are firing because of your epilepsy. And if the neuron fires and the downstream neuron has what kind of connection? Excitatory synapse. So then the downstream neuron will also tend to fire. So the signal will transmit, unless you have inhibitory neuron somewhere. But for all the excitatory pathways, and then maybe there is a pathway from the temporal lobe to the amygdala. So that will also lead to firing of neurons in the amygdala. And the temporal lobe firing is all chaotic because it's coming out of seizures uncontrolled. And the pathways to amygdala would also be multiple, right? And that could lead to firing of neurons in different areas in amygdala. And amygdala is a region involved in fear, processing of fear and also emotions. So that could possibly explain some of these mood swings. Right? And our neural systems have this property that if certain areas you use more, then those connections get strengthened. Right? And if you use less, those connections become weaker. So as a result of this repeated firing and the epilepsy, that connection, the contact between emotional areas, and particularly amygdala and the temporal lobe, probably becomes very strong. And that could explain some of the feelings that he had, right? Emotional state, right? Deep emotional connect or whatever he's having. Okay? What else?
So, in the end, I think Ramachandran was also talking about the possible treatment and its ethics.
Okay.
That if we treat this person, are we robbing him of his spiritual beliefs or something like that?
No, but basically, the patient says that some of the patients say that after this episode, they experience such overwhelming emotions that they might not choose to get rid of the epilepsy just because how good the emotions feel.
How good the emotions feel, how much of a connect they feel with the rest of the world. And in fact, John also says that, right? The reporter asked him this question, and he says that, I don't want to. I'm willing to take all that pain that comes because of this, those moments that I feel connected to the world. Right? That's very interesting, and therefore the ethical issues also show up, right? That is it an illness or is it a gift that you have, right? That he had. And then if you do some kind of treatment, then are you robbing them of this gift? So that's something very, very interesting. Yeah. Okay? So there's some recent, research that says that they have found a God spot in the brain. Inside the brain, and it's in the temporal lobe, somewhere in the temporal lobe only. And we have these systems where you can selectively stimulate certain part of the brain. It's called the transcranial magnetic stimulation. So, magnetic coil, it sends very focused magnetic signals. And once it focuses in some part of the brain, because of that signal, the neurons begin to fire. So without doing any surgery, without going to the Penfield way, right, where you actually touch it with electrodes, non-invasively, just by this device, you can actually stimulate different parts of the brain. And they have observed very funny things, interesting things. And this spot, they stimulate the God spot. You get something very similar to what he got. So that brings us to a very interesting philosophical question. We will not spend too much time on this, but anybody know this guy? Actually, Guru of Swami Vivekananda, Swami Ramakrishna Paramahans in West Bengal, right? And you can see in this picture, he's in a state of trance, right? So his eyes are kind of closed. There's joy in his face. Fingers are in some very special positions. And you look at the expression of people around him, right? They're very curiously looking at him, right? He himself has no, awareness of the outside world. He is what they call samadhi in the yogic literature. And very similar to the trance state that this guy, John, gets into. In fact, if you read the accounts of people like John and accounts of experiences of Ramakrishna, there's a lot of similarities out there. It's spontaneous. It doesn't matter, you don't know when it will happen, it just happens on its own, in his case, and then you just go into that state. The difference is, key differences, in case of John, it was even the triggering of that experience was spontaneous. But in the case of Ramakrishna, he spent years of practice, some yogic practices. And through that practices, he could actually get into those states. But he did not have epileptic seizures. He would have, trance kind of states, but it did impair his workings. He was not able to work. He was employed in a temple as a priest, and he was not able to work as a priest because, all of a sudden he would just stop doing his work he's supposed to do. Right? He's supposed to do some pujas at some point, designated time. And at that time, if he is into that mood, he'll just leave everything. He'll just say, okay, forget it. I'm just going to enjoy this state. Or, he will not even say that. He'll just go into the trance state. But he didn't have to drive a car, right? Fortunately. And he didn't have medical doctors around him. Otherwise, they might have operated on his head and done something. But interestingly, if you read about him, there was his disciple was Vivekananda, Swami Vivekananda, that I guess many people know, right? Everybody knows about Vivekananda? And he was an intellectual. And he used to tell him that, whatever you are doing is just hallucination, you have a problem with your brain. And Ramakrishna would not initially got confused. He said, oh, is that true? But later, he was convinced that no, that's not the case. And what I'm seeing is the truth, is the reality. It's the only truth in the world. Everybody else is hallucinating. That was his view. And finally, he was able to convince Vivekananda as well. So there is a story, you can read about it in Gospel or some writings of Vivekananda. But, I was in Kerala in a hospital doing an experiment on another yogi. He gave a talk here about last week. And we were putting, electrodes on his head, tying it up with tapes and everything. You want to see his brain waves and all of that. And it was, I think they were epilepsy, they were treating epilepsy. And the statement he made was, very small difference between a yogi and a lunatic or a madman. Very, very thin difference. But there is a difference. And that is something which is the most interesting, saying. Anyway, so yeah. So does the brain have a spot for God? Right? Many people say, I've seen God. Right? Does it have a spot for God? Does it have a spot for samadhi? Right? These very high advanced states in the yoga that we talk about. I mean, can they be also elicited by, stimulating a certain part of the brain? And if that is the case, what is that part? Right? So, those are the kind of questions that we would sort of ask you to think about. Right? And something which is probably not very addictive, right? So, you get into a machine, you have a very long day, tired day, you put this helmet on, switch on, 5 minutes, and you are happy again. You had a fight with somebody, you're very upset, put the cap on, switch on the machine, it stimulates the right places, and you are back in the mood, right? You lost a job, wear the hat, switch on, 5 minutes, you are done, right? You are happy, back, right? So if you can actually learn how to stimulate those specific centers of the brain, then maybe you don't need to join IITs anymore, right? Do you? I mean, yeah. Anyway, so we'll stop here. Yes, this one, future devices, you remember?
Yes.
Okay. Tell me. Mary Lou Jaspin.
She started the talk by saying that she has had a brain surgery.
But she's a neuroscientist, is it? She was a
He was an
neuroscientist.
engineer, then he turned.
Engineer, then turned into neuroscientist after the surgery. Okay.
So she had a brain surgery,
Hmm.
and after which for 18 years she has to take multiple medicines.
Hmm.
So she was trying to figure out the exact dosage of the medicine that will help her recover the fastest.
Hmm.
And that is what
Help her recover, not the fastest, but just recover, no? Just feel normal.
Because whatever she does not, I don't think she mentions exactly what happened, but whatever happened did not have a already prescribed medicinal dosage. So she was trying to find it out, and she experimented with which is the optimal dosage, and found out that how minimal changes in dosage can actually result in very big changes in behavior.
In feeling and behavior, right? In thoughts, feelings, and behaviors. Hmm? Absolutely. Wonderful.
So then her main aim was to get back her intelligence and her creative flow. So after this she goes on to mention how the concept of mental images are very important to her and to her thoughts, to her creative flow,
Hmm.
and how she figures out things.
Okay, but let's stop here. Before going further, let's stop here. Can you relate it to anything that we have covered in the dose so far? Small changes in the doses can lead to significant changes in the thoughts and emotions and behavior.
Effect of neurotransmitters.
Neurotransmitters. Those medicines are actually impacting the neurotransmitters, and depending on which medicine you take in what amount, the neurotransmitters in that part of the brain will actually change, and their concentration will change, and therefore the activity in those areas will change.
I think she was directly taking in neurotransmitters.
Oh, directly taking it. But
she was saying that cognitive abilities have been improved or personality has changed. That's what she collected from that experiences. Her personality is also changed and cognitive ability also. That's why she perceived that brain interface that I should go in the images of this. What is happening now?
what is happening in the
She
brain?
uses, she takes.
But neurotransmitters will not go directly, right? They have to cross the blood-brain barrier somehow.
That's what she said. She's directly taking in different doses, not even direct doses, combinations. This one time she took the combination of neurotransmitters that a 22-year-old man would take. And
Yeah.
she explains about her experience.
That's right. Feeling that you are the, what is the thing?
Yeah, because Mary was required to take neurotransmitters externally.
External, but not inject, but orally take.
Maybe orally, but
I
it
see.
was neurotransmitters.
I see, neurotransmitters only. Yeah, surgery in the part of the brain that produces neurotransmitters, and sense of self itself changes. Right? And sense of who am I, and thinking, and then the behavior towards people. And she tried to adjust the doses to get the intelligence back to thought flows. Right? And now this is a life-changing experience that she had, because all our lives we are in, we believe ourselves to be like XXX, right? Certain type of person with certain personality. But that is nothing but the balance of neurotransmitters in our brain, which can be altered. In her case, because of this, surgery, it altered, and then because of the medicines that you took. But are there other methods to alter it? The relative concentration of neurotransmitters in different parts of the brain. So,
Show that activities like exercise increase certain.
exercise, right? If you do exercise, then the certain neurotransmitters change. Right? Definitely, it happens. Right? What? Huh? Psychedelics have also, also sort of changed the neurotransmitter levels. Alcohol as well, yes. Sometimes good food also. Right? It releases alcohol in the blood.
Pretty much anything, I guess.
Huh? Yeah, but the impact is different at different things. But
Even in now had a study by people they have shown the gamma stages due to yoga practices.
yoga practices, that's also very interesting, right? Yoga and meditation practices have also shown to change the neurotransmitter levels. And yeah. So essentially, so if you look at the synapse in the boundaries between the dendrite and the axon, the neurotransmitters that modulate the flow of action potential between the cells. And if you can change it, very substantial changes can happen. And in the earlier part of the lecture, of the course, I talked about, so many mental health problems the world is facing. Right? And today we had a neurosurgeon visitor from Australia, and he was talking about this thing, that people are having so many issues, right? Especially elderly people, Parkinson's and all of that they're having. And we are looking for solutions in the brain, that, what is it has gone bad. But essentially these neurotransmitters that have changed, why have they changed? Their concentrations, why have they become different from the normals? It may be because we are consuming food which is altered, right? Either genetically modified or by pesticides and chemicals. And over the years, we have been doing it for all our lives, right? 30, 40 years. And then that has, one-time food, maybe the effect is not there, but over a long period of time, maybe that is what is causing this thing. So, we don't know the answer to this, but it requires a very substantial study. Right? We have been able to figure out how to increase the productivity. We have been also able to figure out how to increase the longevity. But we have not been able to figure out how to improve the quality of life, the subjective quality of life. Right? So we need to think deeply and look deeply into these issues. So, yeah. And thus, there are different types of neurotransmitters. They play a very important role. And we have, interestingly, our body has developed mechanism so that they stay within the normal range. So even if something goes wrong a little bit, the body can adapt. And it can also, produce extra if something is deficient, until it actually causes a threshold. So that is the problem, because, if you have something, if you inject, a food with some kind of poison in it, you will not even realize it, because your body has defense mechanisms. Until, you keep on injecting it over and over again for a long period of time. And then body begins to give up. But okay, I can't do this anymore. So, this is a very important area of research and a very difficult one too, because there are lots of opposing forces that come in the way when you do this, when you actually try to do this. But absolutely very important to actually look at these long-term impacts of these, especially given that the mental health situation is going really downhill for all over the world. Okay? So let's come to mental images. Yeah. So then she actually started to think what's happening inside our brains, right? To want to know. It's okay, you don't have to wait, it'll come.
Like her perception was actually like because of the intake of those drugs, her perception was altering, and
Okay.
so was the mental images that she was generating in her
Hmm.
mind. Then she had an idea that mental images equals to ideas, and
And then she talks about a very interesting experiment, right, with fMRI.
yes,
Mental images versus ideas.
yes.
So what was it?
So basically, what was happening is that seeing the actual thing and while imagining it, they took fMRI data of both, and when subtracted the data, it was almost around zero. So viewing and imagining it almost had a similar effect.
Yeah.
So the same regions, for example, if you're seeing something of occipital lobes, like some part of occipital lobe will fire.
Will get activated, yes.
If we are thinking about the same thing,
Yes.
the same part of the occipital lobe
Would start getting. So that is very interesting now. You are having an experience. Right? And why are you having that experience? Let's say you touch something hot. You got an experience of, hot being there. But why do you get that experience? Do you get that experience because something hot touches here? Or do you get that experience that the signal that something hot, the information that something hot touched here gets related to the brain? The second one, right? Because if you sort of cut the nerves that are carrying that signal, then you put something hot here, nothing will happen, right? You will not have that experience. So, if you are having an experience, it's because those areas in the brain, the neurons have actually become active, which is, it starts from this and then the signal gets communicated. But now if you imagine the same phenomena happening, the same neurons will get activated in a very similar manner. Right? So, then again philosophical question. What is it? What is the reality? That what you see, that what you imagine, right? Many people hallucinate, right? Schizophrenia. How many people have watched The Beautiful Mind? Right? So you all of you, okay, wonderful. Most of you actually. So this is a story of a Nobel laureate, right, who went into schizophrenia, right? He got a Nobel Prize. John Nash. Biography of John Nash.
Founder of game theory. Was the founder of game theory.
Founder of game theory, yes, absolutely. He actually gave a talk in the seminar hall in 2003, I think. We invited him at IIT Delhi, and he gave a talk, here. And okay, let me pause it. For them, they see it. Although people around them don't see that thing, but they see it. So for them it is very real, because most probably the corresponding neurons in their brains are actually firing. And that's what is giving that experience to them. But then how do we...
Schizophrenic patients, the dopamine is more, the neurotransmitter dopamine. Because of that, they go to this kind of hallucination or other kind of symptom, delusional hallucination, increased dopamine. So even from the yogic perspective, we say if you do a closed eye meditation, that gives us same kind of dopamine release. So you should avoid with these kind of patients, such kind of practice.
I see. So if you do closed eyes practice, closed eyes meditation, it leads to dopamine increase.
So you should not be doing closed eye meditation. person who is having such kind of symptoms.
Who is hallucinating? Oh, interesting. Yeah. And then trauma is another example, right? Where you had some experience in the past and now you cannot come out of it because you imagine that thing, you remember those things, you recall those experience, and then you start having the similar experiences. And that's a big problem for them, right? That makes them very, optimally functional. So two issues, right? What is the truth? What that hallucinating person is seeing or what you are seeing versus not. Right? What is the reality? And second thing is, if you say that what a person hallucinating is seeing is not real, then how do you get them to see the real things? Okay, what else? Yeah, so this is the picture that she had drawn, and here you can see the occipital cortex. So this is a perception of an object, and this area is getting activated, while imagining same area is getting activated. And this they have designed using fMRI, right? This is an fMRI based experiment. Okay, what else?
Another study
where they reconstructed the visual.
They reconstruct the visual, right? So use AI and ML, using that they reconstruct the. And this represented, right? This I showed you earlier also. So, and it is pretty not bad at all, right? At least for layperson, it's kind of. And then she says that advances in fMRI technology is happening, and if we up the resolution, then we can communicate directly with human thought. And we are getting there, right? The other video that I showed you, where this person was watching, and then the thoughts were being shown as subtitles. That actually is a step in that direction only. So how do we increase the MR resolution? Essentially, we are right now at generation 3. So we already got in 2 generations. The first generation was, bigger magnets, better magnets, with, much more uniform magnetic fields. A lot of engineering, a lot of physics to make sure that the magnetic field is uniform, a stronger magnetic field. So, we started with, half a Tesla, then, 1 Tesla, 1.5. Now we have 3 Tesla is almost standard. Even 7 Tesla fMRIs are being built, MRI machines are being built for experimental purposes. 9 Tesla also has already been built. But industry standard right now is 3 Tesla. Right? Then the second generation employed this technology called multi-coil. And, basically in the magnet there are these coils that measure the signal, and instead of a single coil you put multiple coils there, and then you have to use some special data processing methods to get the signal out of that, to reconstruct the images. So that also is very well understood. Many people have worked on this, and now we are looking at third generation where more advanced signal processing technique and machine learning is being used to increase the resolution even further. So, yeah, okay. Anyway, so this one, nobody has gone through it. Okay, so we'll do it next time. Okay, so then, as we talked about, what is functional MRI? It's a 3D movie of the brain, and it's an indirect measure of brain activity that we already talked about. This is a typical fMRI setup, where the person lies down in the scanner while the data is being recorded, and you can give different types of inputs. Auditory inputs, visual input through an MR compatible screen, mostly a mirror, and then joystick and keyboard inputs. Okay. And what you get is a time series data. You have already seen, right? You've already written a code to hopefully all the data science people to look at this data. Now, what are the properties of this data? The thing is that a signal is very weak. And what do we mean by this? So you would look at a particular voxel, you will see what the value is. Maybe it will turn out to be 5,000, 6,000, 10,000, right? But that is your MRI signal, right? Now the functional part, what is the change in that signal due to change in the blood oxygenation level? That is hardly 1 or 2% of this. Alright, so in your assignment you have to find the visual cortex and the voxel that is responding to that, right, which is changing. So if your signal is 5000, the 1 or 2% would be about 50, the change in the voxel values because of the stimulus. So that is what you will observe, right? Signal is very noisy, is very weak. Other thing is very noisy. So, you would see a signal of 5000, it will have these random fluctuations, and then maybe, because the checkerboard has been presented, it will sort of, from here maybe it will increase, but still it will be very noisy and then it will come down. I'm sort of, excuse my drawing, it's not like this, but, once you try to plot it, you will see. It's already very noisy. And the signal to noise ratio is very low. Okay? So then, how do we figure out the brain function with this? Because you can't be sure. It could be just noise. How do you know whether it's signal or not? So you have to do a lot of repetitions. So if you look at this data that we have given you, 3 repetitions are there. But typically you would do 5 or 10 repetitions of the same stimulus. And then you would actually get the signal out. And you need advanced statistical methods to make sure that whatever conclusions you draw, they are valid. They are not because of statistical noise. They are not because of random noise. So that we will do in much more detail in the later half of the course. The statistics part we will cover. But the other problem is that this data is very large. One brain has 100,000 of voxels, and one subject has, depending upon the length of the scan, either 100 or 500,000 time points. And then you have to repeat the experiment on multiple subjects. Right? So maybe you would have a typical experiment, you would have 100 subjects. There's some human connectome project, they had thousands of subjects. So, to draw meaningful conclusions, you have to repeat the same experiment on multiple subjects. So if you combine all of this together, thousands of subjects, millions of voxels, and then thousands of time points, the data becomes very, very large. And you have to process it. So it's a big data problem also. And of course, you cannot just throw a software at it, process the data and get the output, because you have to interpret. And while you are interpreting the data, you have to understand the neuroscience, you have to understand how the fMRI works. Because you could make incorrect conclusions. Right? I was processing, I was getting some data of some, again, schizophrenia patients. And just out of fun, I had their demographic data as well. So just out of fun, I thought that I'd compare whether the male brain is different from female brains or not. So I did that study, and I found that, the female brain was 10% more brighter than the male brains. And, I thought I made a wonderful discovery. But it turns out that that is not true. Right? So the female brain does not have 10% extra signal. What actually is that the female brain is a little shorter than the male brain, a little smaller than the male brain. And then when I was doing the processing, it was registering everything to the same template. And it was some kind of doing the normalization. And because of that small size, the intensity value increased in the female brain. So it was an artifact of data processing, not a real thing. So if you do some analysis, some machine learning, you cannot just blindly say, oh, I'm getting this. You have to look through each and every step very carefully to see whether those conclusions are meaningful or not. Okay? So the BOLD, we already talked about blood oxygenation level dependent signal, right? That's why it is called BOLD. And it does not measure the neuronal activity directly. It measures the oxygen, or change in the oxygen levels, or the ratio of oxygenated to deoxygenated hemoglobin in the blood. And it's a very weak signal. And since this signal does not really depend on the neural activity directly, it is not directly correlated with the neural activity. It is delayed through what we call a hemodynamic response function. So I'll come to that in a minute. Oh, okay, this slide is already talking about that. So let's say you have an fMRI experiment, the subject is inside the scanner, and you apply stimulus from here to here. Right? So let's say this is time t equal to 1, 2, 3, and 4. So at time t equal to 1 to 3, you apply some stimulus. Right? So what do you expect when you apply the stimulus? Let's say it's a visual stimulus. So you expect, your neurons in the visual cortex to begin to fire, right? Begin to become active. So you expect the signal to be there. But actually what you see is instead of the signal increasing, there is an initial dip. Okay? And then you see after a little while, after maybe half a second or after maybe 1 second, then the signal reaches a peak. Okay? And when you remove the stimulus, you expect the neurons to stop firing. And they will indeed stop firing. But in the bold signal, what you will see is it will continue to be high. And it will come down only after some delay. And it will not even come down to zero, it will actually come down to a negative value, which is called the post-stimulus undershoot. So we have initial dip in the bold signal after the neurons in that area become active. Then we have what are called an overshoot, where the, it stays higher than for the most of the period. Then it's a positive bold response, this range, and then there is a persistent bold activity even after the stimulus has been removed, and then there is post-stimulus undershoot. So the signal that you get does not directly depend on the neuronal activity, it depends through this very complicated thing. Okay? This is very important to understand in the fMRI processing. Okay?
So why this post-stimulus undershoot is there any specific reason for that?
Yeah. So basically, if you look at the neuronal activity, okay, how the neurons fire, that activity scientists have isolated a single neuron from maybe squid or from other animals, put it in the lab, put it on a slide, engineered intracellular engineered single unit recordings, and they apply various types of stimulus and they try to see how it behaves. Right? So this part we kind of understand very well, that when you apply the stimulus, how the neurons will react, and a lot of work has already happened. Now here when we are doing MR scanning, how the magnetic field changes, how does it give rise to the bold signal, all of that is MR physics, right? And that is also reasonably well understood. Okay? But this particular issue that we talk about, which is that in a particular region the neurons start getting activated, right? They start firing. Now what happens is when the neurons start firing, they will require oxygen, right? So oxygen is in the blood, so then the blood supply to that area has to be increased. So initially what happens is because of that oxygen consumption there is a dip. But then the brain actually supplies the blood there and it actually overcompensates. So this dip is because of initial oxygen consumption, and then it's an oversupply of blood, and that's why we get the positive this one. This one I'm not sure, but I think there is some work to kind of understand why this happens. But this whole mechanism of the neurovascular coupling is called a neurovascular coupling, because nervous system, the neurons are firing, that is one system, and now the blood flow in that area is increasing. That's a circulatory system. It's a different system, different mechanism. And this coupling is not very well understood even today by the scientists. Okay? And yeah. So this is modeled by what is called a hemodynamic response. We'll come to that in a minute. But it is modeled based on the observations only. In the sense, empirical observation. It's like, there's some pattern you're observing and you fit a function to the pattern and you say that, okay, it is this function. It's not governed by, very solid understanding of the underlying mechanisms there. Okay? So I'll kind of, I'll talk about it in more detail in the subsequent slides. But before we come there, now you kind of understand the whole mechanism of this fMRI, right? So you'll get something, some signal will come. Now what do you do with this? Right? What are the type of data analysis you can do once you have that data? So first type of analysis is what we call functional brain mapping. What do you mean by this? What part of the brain is involved in a specific function? So vision. What part of the brain is involved in vision? Right? We know occipital cortex, but now you can ask more interesting questions, more specialized questions. For example, if I see vertical lines, then what part of my visual cortex is involved versus I see horizontal lines or I see slanted lines, right? Or if I see motion, then what happens, right? What part of the brains are involved in those? So just vision alone is such a large field, right? Because vision would also entail object recognition, face recognition. What else is involved in vision?
Depth perception.
Depth perception. What else? Spatial orientation, right? The map of the region around me, right? So all those are, in fact, I think there was a recent discovery of what are called the place cells in the rat. So you put the rat inside this chamber, and then very specific cells get activated in the hippocampus or some regions. They are called the place cells. When he visits that particular place, only then those neurons will fire. So that discovery was made recently. So like that, in the humans also, are there, circuits like that? Orientation in the space or position in the space. So all of this would fall into what is called functional brain mapping. What is it saying? What kind of questions we are asking? Which part of the brain is involved in which particular activity or specific function? And this presupposes. What does it presuppose? Localization, right? The brain function is localized, right? So one region is actually involved or, in processing one specific function. Then there are, these. So what happened was when the fMRI and all came in, we thought that, we'll be able to handle mental illnesses very easily. Because now we can look inside the brain, we don't have to operate upon this. And person with depression, person with Alzheimer's, person with Parkinson, we can just do the brain scan and from that figure out. Or some person with some kind of illnesses, we can just do a scan, figure out what's wrong with it. But unfortunately it was a failure. We could not even distinguish a depressed individual from, somebody with, let's say, hyperactivity disorder, attention ADHD or something of that sort. And that led to a lot of studies on what we call functional connectivity studies. And what does this mean? That how do different brain regions interact with each other? So here we are not talking about function of this region. What we are saying is, how do two specific regions interact with each other? Can you think of an example?
The Broca's area is involved in memory production, and we would need the motor cortex to make the particular tongue movements to produce those particular whatever we are trying to speak.
Yeah.
The Capgras delusion, the connection between amygdala and fusiform, I guess.
Yeah, but let him finish, then we'll come to you. So then
Right, so the motor cortex would have to
coordinate with the Broca's area to actually produce that effect. Yes, absolutely. And in case that connectivity is broken, then it may not function in a normal manner. Here you have to distinguish between structural connectivity and functional connectivity. Similarly, when we talk about functional brain mapping and anatomical mapping, right? So where you are looking at the structure. What is the structure? Now, when you talk about connectivity between Broca's area and, let's say, the motor cortex, or the areas for moving the mouth, you can already begin to look at the structure of the fiber tracts, the neurons. But when we talk about functional connectivity, what we are saying is whether the neurons in those areas are getting simultaneously activated. Right? When the Broca's area neurons get activated, then do the motor area neurons also get activated? Right? Or with a delay, maybe. Right? So that is the difference between structural and functional connectivity. Okay? Yes, you were saying something.
Yeah, I was also another example is that for face recognition, the connection must be between the fusiform gyrus.
Yeah, so the connection between the fusiform gyrus and the amygdala, according to Ramachandran, is broken. Right? How do you test that hypothesis with fMRI, with functional MRI? What you could do is, You could get a healthy, normal person inside the scanner and show him or her picture of him or her mother. And you see what areas get activated when that picture is presented. There will be visual area, there will be face recognition areas, face areas, but maybe amygdala will also get activated. Right? And to do the same experiment with a person with a cap brass delusion. And then you see whether amygdala activity is still there or not. And that's how one of the methods to actually put some evidence in this theory of Ramachandran. Right? So basically, yes, when we talk about interaction of different brain regions, one way of figuring this out is simultaneous activity. And how do we do that mathematically? Correlations, right? You can look at the correlation between two different voxel activities, right? But you can look at many other things. Correlation is the simplest thing. You might want to say, after this area gets activated, does that area get activated? But how much after? Maybe 1 second after that, or 2 seconds after that. So then you can look at delayed correlations. You delay one signal by 1 second, then are they correlated or not? Right? And other very interesting thing is effective connectivity. Okay? So imagine you have a voxel or a brain area. So let's say this is V1, this is V2, this is V3. So there is a connectivity, a structural connectivity from V1 to V2 and V1 to V3. Now, when you collect the data, you will see that whenever V1 is active, after that V2 becomes active. And also V3 becomes active. Right? Because this region, the neurons in this region are actually activating the neurons in V2 and V3. Okay? Now when you look at the data and you find the correlation between the activity of V2 and V3, you will find a high correlation between activity of V2 and V3. But they are not structurally connected to each other. Right? So what we want to see is this word cause. You want to see a causal pattern of connectivity. Whether this region actually causes activity in the other region. Whether activity in region 1 causes the activity in region 2. We are not only interested in correlations. So those kind of studies are called effective connectivity studies. And there are mathematical models for causation and all of that. So we'll not get time to cover this in our course. We'll do functional connectivity briefly. Most of the course will focus on functional brain mapping, the required statistics behind that. And yeah. But now coming to multi-voxel pattern analysis, so essentially, yeah.
So what is the difference between functional and effective connectivity studies?
Functional connectivity is saying that, when the two regions are active simultaneously, do they interact or do they get activated simultaneously most of the time? So, for example, you look at the blood sugar level of a person. I'm just giving you a totally different example, but it's a difference between correlation and causation. Right? So functional connectivity talks about correlation, and effective connectivity talks about causation. So, for example, if you measure the average blood sugar level of a person, and also look at the diabetes medicine intake, maybe what you will find is people with high sugar also take diabetes medicine. Right? So the intake of medication would be correlated with the blood sugar level in that person. Does that mean that the diabetes medicine increase the blood sugar level? No, it's actually opposite. So there may be a correlation, but it's not a causal relationship. Taking that medicine does not cause the blood sugar level to rise. If you want to understand the causation, then you have to see that when you give that medicine, after that what happens, and when you stop giving that medicine, after that what happens. So in the same manner, if I see that these two areas, V2 and V3, are highly correlated, the activity in these areas are highly correlated, then we can say that they are functionally connected. But I want to see whether they are effectively connected or not. Whether the activity in V2 will cause the activity in V3, or whether it will be the other way around. That activity in V3 will cause the activity in V2, or none of these, right? Not related at all. So V2 and V3 are independently active. So that is done by these effective connectivity analysis methods.
So basically, it is the next step or a deeper step to functional
That's
activities.
That's right. Yeah.
Fundamentally, if MRI is taking a surrogate measurement of that oxygen, so it's not taking directly the measurement of the neurons at all, right? So how effective can it be for telling that this causes this region to activate? Like in the EEG, MEG, we are taking directly the action potential value by EEG and MEG. But for this, we are taking a surrogate value, and that surrogate value is talking about the causation, like.
Yeah. Very good question. So first of all, in EEG we are not taking the action potential value, right? We are just taking some electrical activity which we believe is caused by action potential, right? But it could be because of action potential of multiple neurons, multiple places aggregated in a very complicated manner. But the good thing is we have very good resolution. We are getting 100 Hertz of a sampling rate. But in fMRI, a sampling rate would be like 1 second or 2 seconds, right? That's very, very small. And besides, you have this hemodynamic delay. When you apply the stimulus, the bolt signal does not change. It takes about a second or two for it to change. And the neural processes act in millisecond range, right? Half a millisecond or maybe 50 or 100 milliseconds. Right? That's the kind of speed at which the action potential propagates from neuron to neuron. So then how can you do effective connectivity analysis of fMRI? And in fact, if you read effective connectivity analysis papers published in the fMRI domain and take them to a neuroscientist who works on neurons, right, who does single unit recording, he is going to throw it out of the garbage. He's saying, I don't know what this means, It doesn't make any sense to me. But yet, there are processes in the brain that actually happen on the time scales of seconds. Like, things involving cognition. Right? Things involving memory, recall, and stuff like that. So there, although, at least we know that, it takes a time scale of like half a second or 1 or 2 seconds. For those kind of processes, then you can actually, look at that data and see whether which region is causing the other or not. Any other comments? Okay. So then what is happening now with the advent of machine learning and all, people are creating those models and based on that doing lots of interesting stuff, lots of interesting data analysis. And this term was used earlier, which is the multi-voxel pattern analysis. So we are not looking at one brain region getting active. We are looking at the patterns of brain activity. So at one point in time, 10 different brain regions may get activated. Right? Then the question is, when does this pattern repeat? And when this pattern is present, what does it mean? Okay? So there have been papers that talk about, for example, visual cortex, right? Visual stimulus presentation. Now, using the fMRI signal of non-visual areas, can you predict when the visual stimulus was presented? And the answer is yes, you can. Right? So then the question arises, our brain information is localized or distributed? Because, we started with the localized. Now when you do machine learning based analysis, you can say that even the visual information you can get from non-visual areas. At least make some predictions of, very simple rudimentary predictions. Now if that is possible, then those areas which are also non-visual have that information somehow. Right? So that opens up a new, completely different worm of, can of worms. But anyway, so I think we do what we do. We do what we know to do. So, many people have applied novel, machine learning methods to be able to try to look at this. But all of these are like blue sky research, right? They are showing these kind of things possible. Like the video that I showed you, which was predicting thought, is also very blue sky. That how is going to get used and what are the actual mechanisms and whether it is actually predicting the thought or not is right now it's all open. When even you reconstruct the brain visual activities, what does it actually say? Can you look at the voxels that are helping you to reconstruct the visual activity? And then based on that, can you understand the brain function better? So those are the kind of problems that we have not yet started looking at it more seriously. And that's where this interaction between data scientists and neuroscientists would be a very important one. And then finally, the dynamical activity. So right now we are talking about all static activity. You show this happens. Okay? You show that happens. But our brain doesn't work like that. I show you a picture of somebody that you hate, and I remove that picture. But that impression is still inside you, right? Your mind will start working and start recalling certain things and all. And now if I show you another picture, your mind is already primed by that. So it's not as if I showed you a stimulus, some neurons fired, I removed the stimulus, those neurons stop firing. It happens. It happens in early processing, right? Early visual processing, early sensory processing areas and all of that, it happens. But when we talk about more complicated processing patterns like emotions, cognition, the sense of self and all of those things, they are a dynamic process. And a single small trigger can actually trigger a chain of thought inside your brain. And how do you uncover that? How do you discover that? From the data, can you figure those things out? And this is a very, very interesting and important area of research, which I don't think people are doing right now. So that's also something to think about. Okay. So again, same thing in pictorial activation analysis. You get a blob in the brain, right? The picture that I showed you earlier. This region in the brain gets activated because of this stimulus. So this region is involved in processing of this stimulus. Okay. Connectivity, what will you get? You will get a graph. This voxel or this node is connected to that node, is not connected to this node, and so on and so forth. So it's very hard to visualize this to begin with, right? Because if you have n voxels here, your output, the result size is n, right? Which is number of voxels. But here it becomes n squared, because every pair of voxels you have a connectivity. And then machine learning, MVPA, we don't even know how to visualize the data. You can make the predictions, that's fine. You can predict or you can reconstruct the image, but then what did we learn about the brain function? That we don't know yet. And spatiotemporal dynamics is a complex interaction with the different brain areas. And how do you model that? How do you take it forward? We don't know. But in this course, we are primarily going to be focused on activation analysis. But I would encourage you guys to, look at some of these things here, come up with, newer models, newer ways of looking at it. And if you can, apply machine learning or those kinds of things on this data and try to come up with some new insights, that will be wonderful. In your projects mostly. Your assignments will be doing mostly activation analysis, okay? So it's used for deciphering human brain function. And what kind of questions we ask? Localization of brain function. Area X in the brain is responsive to task Y. Right? So that's the kind of questions we're asking and answering. But this also can be multiple subtle things, no? So area X is responsive to task Y. Area X is selectively responsive to task Y. And area X is more responsive to task Y as compared to task Z. What's the difference between these three statements?
What?
Let's start with X, statement number 1 and 2, right? 1 and 2. Area X is responsive to task Y. What does it mean?
Some
correlation.
So every presentation of task Y,
Huh.
the area X gets activated.
Okay.
But then for the second one,
Wait. So let's look at it a little carefully. So Y is a stimulus, right? A task or something, right? It could be like showing a picture of your mother, right? That's your stimulus. So whenever that stimulus is presented, it need not be a passive stimulus. It could be an active thing also. It could be, you're supposed to imagine something. So no stimulus is presented, just a cue presented, and then when you see that cue, the subject is supposed to imagine something. Or the subject may be asked to move the hand when something happens, move the right hand, for example, right? So it's a task. That's why we don't call it a stimulus, we call it a task. So when the task Y happens, which area X is responding to that? Means it gets activated. The neurons start firing, which we decipher indirectly, but in reality what we see is the bold activity there increased, the bold signal increased when the task Y was presented. Okay, fine. Now let's look at the second one. Area X is selectively responsive to task Y.
So only for certain stimuli of task Y, the brain area X is getting activated.
What do you mean by certain stimulus of task Y?
So can it be that multiple area X only responds to task Y as a whole and not certain stimuli of task Y?
Or maybe it only responds to task by nothing else.
That would
Can that happen that one area is only for one task and nothing else?
Probably not. This is a question.
Yeah.
I think selectively responsive might be that there are multiple areas being activated.
Yeah. So here selectively actually generally refers to the task. So what it means is that it responds to task Y, but only to task Y, no other task, which is sufficiently different from task Y. Right? So, and not to task Z1, Z2 and so on, or not to other tasks. It only selectively responds to that task only. Okay? And that you will see. This particular example you will see in your next test, Dr. Nancy Conveser. She discovers this area which is selectively responsive to the perception of human faces. Fusiform area, yeah. Fusiform face area. Okay. So now finally number 3, area X is more responsive to task Y as compared to task Z. There are 2 tasks, and now what we are saying is which of the tasks gives you more response in that area, right? So either we can say, it does not respond to any of them, it responds to both of them, but in case it responds to both of them, then to which of them it responds more. So that is what we want to find out. So those are the kind of studies that we will do, the kind of questions we will answer in doing the localization of the brain function. But this, if you do, you only do it for one subject, right? You get data for one subject, you do it, but then you have to generalize it to population. So area X is selectively responsive to task Y in this group. Right? So how do you do that? How do you figure that out? Maybe when I present a stimulus to you, some area gets activated, which is also selectively activated. But it only happens in you, nobody else. So that is not of much value. You want to generalize it to a population. Okay? So that's the other kind of study. And then you can also say area X is more responsive to task Y for patients with this particular condition as compared to healthy subjects. So now you have a contrast. Right? A contrast between 2 different tasks, task Y and other tasks, and a contrast between subjects, which have illness and which don't have illness.
But the task is same.
The task or the pair of the task is the same. Yeah. Okay? Is clear? Okay. So right now I'm putting it in words, but then I'll need like 10 lectures to put it in the maths. Right? So, yeah. So what is the result of all of this? An activation map. What do you call a brain activation map? Typically we put it like this, color coded. So here is the brain. You would have seen, right? You can browse through this, you have the fMRI data. And then we color code it. So these colors, I can choose any colors. These don't mean anything. It's my choice. Right? What I have is an activation map. So what is the probability of activation? That is what I get. And depending upon the threshold, I can give them different colors. So when you see this picture by Jaspin, Mary, right? This one here, right? So this thing, what you see, is an activation map. Which area is getting activated? And that is the result of the analysis that we do after this. And there's nothing red out there. You can just pick any color there just to represent the probability values. Okay? Okay. So, yeah. So this is what you will get in the end. What is the function of area x of the brain, right? That is also, you can reverse the question. Initially you are asking for this task, which area is activated when you do this task. Now I am saying, picking this area, what is the function of this area? What does it do? Right? And then, yeah, the other thing is which areas are involved in task y. Okay. And you understand the difference between this, right? Which brain areas are involved in task y? What does it mean?
So what areas get activated during the task y?
Yeah, when the task y is presented, when the task y is there, which brain areas get activated? Or in other words, which brain areas have higher bold signal as compared to the baseline when task y is presented? Which is the same as, which areas respond to task y. It is the same as that. Which areas respond more to task y as compared to task z? So you are looking at the change in the bold signal with respect to task y and also with respect to z, and then subtract the two. Then you will get that. Which brain areas selectively respond to task y? How will you figure that out?
That it only activates if task y is given, like if and only if it.
No, not if and only if. Only if. No if.
But it's selectively.
Selectively.
It's not supposed to.
Yeah. So it responds to task y, but not to other tasks like Z1, Z2, Z3, Z4. So you need to have.
It has to be
Huh? Okay, yeah. It looks okay, yeah, that looks correct. So anyway, so now if you respond to task y and only to task y, then you need to have multiple tasks, right? Z1, Z2 and so on. And then you are seeing that, whenever the task y is present, that area is getting activated. But in none of these tasks, that area is getting activated. Right? And then you have to generalize it to the population also. So that's what you have to do. Okay. So before I stop, you have to do an experiment design before you can do these kind of studies. And in the experiment design, you were going to design your stimulus, you're going to design your task presentation protocol so that you try to answer the question, and then there are a lot of things that you have to take care of. So that we'll discuss maybe in the next class. So we'll stop here today and then we'll continue on Monday. Okay, thank you.